This is Naveen Duhan. Today I'm here to present my tool pySeqRNA, that is an automated Python package for NGS data analysis. Our main objective was to develop a Python package which can analyze NGS data from end to end and which also supports job scheduling using Slurm on high performance computing clusters, and as well as manages memory and processes on local computers. Before executing pySeqRNA, user needs to provide a input file in the following format, like the sample name, replication, identifier, and the file. In case of paired end data, user can add another column, that is file 2. pySeqRNA processes RNA-seq data in two phases. In first phase, it only count reads which are uniquely mapped to a single gene, and the reads which are not assigned uniquely to a single gene in the phase 1 are further processed in phase 2, and these reads are assigned to multi-map gene groups. And by using this, we can extract the multi-map gene information directly from the data rather than relying on the annotation. In our test on multiple datasets, we found many groups represent known relationships. For example, paralogous genes. In pySeqRNA, by default, MMGs are counted in such a way that a gene should be present in at least 100 reads and 50% of the samples. Currently, pySeqRNA supports miRNA-seq and RNA-seq. A user can provide input sample, mandatory arguments like reference genome, feature file, and in optional arguments like which tool to use. And it takes those information from the user. And the parameters for different tools, different scripts are taken from the pySeqRNA package. And pySeqRNA store intermediate results in dictionaries and proceed further. Ultimately, all the results are saved in a results directory with all the visualization and a HTML report. MMG groups are the novel feature of this pipeline in that, for example, suppose we have 7 reads and 3 genes. In this figure, read 1 to 5 mapped to gene A, and read 4 to 6 mapped to gene B, and read 2, 4, and 6 mapped to gene C, and read 7 is unmapped. So when we count them, so read 1 is mapped only to A, that is uniquely mapped read. Read 2 is mapped to A and C, that means it is a multi-map read. So similarly, read 3 is a uniquely mapped read, read 4 is a multi-mapped, read 5 is multi-map, and read 6 is also multi-mapped. So when we count MMGs based on the default parameters, it comes to be 2, 1, and 1. MMG 1 is A and C because it came twice. Similarly, A and B came once, A, B, C also came once. And there are 3 scenarios how we count or disregard multi-map reads. In first scenario, the read is 100% matched on both the genes. Then it is considered in the MMG counts. The other scenario is for one gene the read is mapping 100%, and on the other gene the only the 70 or 80% part of the other gene is of the read is mapping on the gene. And in third case, one read is mapping 100% on gene A, but on gene B only a fraction matches. In that case, we disregard those multi-map reads. After this, pySeqRNA also supports functional enrichment analysis like gene ontology and KEGG pathway analysis for more than 200 animal and 120 plant species with beautiful visualization or publication ready pictures. You can download pySeqRNA from GitHub or for any further questions, you can contact on those emails. Thank you very much. If you have any questions, please drop down them in the comments. Thank you.